when we feed the animal, we're really feeding the microbial ecosystem. And the ecosystem accounts for the ability of the animal to ingest fibrous feedstuffs and convert them into high quality, humanly desirable and humanly edible protein and energy sources. So the fibrolytic capability is core to the role of the ruminant in human food production. And although we presently pass a lot of starch through these animals, it could be in the future that the role of, well, I will say that in the future, the role of the ruminant is stable because it will always be able to convert not only starches, but also fibrous feeds into humanly edible food.
Welcome to the Beef Podcast Show, brought to you by Wisenetics. I'm Dr. AJ Tarpoff, and we are dedicated to bringing you the latest insights and discussions from the cattle industry. So sit back, relax, and get ready to expand your knowledge with today's episode. And really fortunate to have Dr. Dan Schaefer to be our guest here at the Beef Podcast Show. To give a little bit of background, it's hard to put a blip for a full, very, very eventful career, but I'll give it a shot. So Dr. Schaefer, Emeritus Professor at University of Wisconsin-Madison, currently sole owner and operator of the Schaefer Beef Consulting. I'll ask you a little bit more about that. But Dr. Schaefer, he was born a farm boy in Wisconsin, which I love to hear that hey, we all have to start somewhere, and it's wonderful where we come from. But from Wisconsin originally, when he started his academic career, he was a faculty member at Purdue University, and then moved over to University of Wisconsin-Madison, where he taught just about everything, from Rumen Microbiology, beef production, career orientation, advised undergrads, mentored graduate students, eventually had moved his way up and was the department chair in animal science from 1999 to 2016. Afterwards, this is really interesting because this is a unique program at University of Wisconsin, but took over the director of meat science and animal biologics discovery program in that new facility that was built, a $57 million building, and where he managed and kind of oversaw that facility until his retirement in 2019. He is a fellow of the American Society of Animal Science, and he's a great guy, and he's welcome to the Beef Podcast Show.
Well, thank you, AJ. It's my pleasure to have been invited to participate in the podcast. I look forward to our conversation.
And we'll get to the conversation and the topic at hand. We're going to dive in a little bit about Rumen Microbiology and the insights so we can use that information to grow our understanding of the beef industry and kind of the productivity of these animals. But tell us a little bit more about what you're doing now. You've been retired for a handful of years, but you can't stay out of the cattle industry, can you?
That is true. So after I retired, I realized that I missed my contact with the beef industry and with cattle people. So I wanted to reinstate that. So at the present time, I am interested in hearing any question that has the word beef or beef cattle in it. Obviously, the feedlot sector is not large up here, though we have some cattle feeding on a smaller scale. But I'm interested in trying to help people with my knowledge. And because I taught beef production and because I'm interested in the system, I'm aiming to provide beef production system solutions.
You almost sound like an extension, specialist.
AJ, what I do is a lot of education. I do develop some spreadsheets also, but I'm providing the same kind of service that Extension provides. It's just that in my case, it's individualized. I'm focused on the question that's posed to me by the person who has some need or problem or is seeking some kind of knowledge.
I love it. I know that type of role and what you do currently. It's fulfilling because you really get to dive in. You get to help people. I applaud you for taking that step.
Thank you.
Surprisingly, even during summer months, cattle don't get enough vitamin D. That's where High D comes in. High D is the gold standard 25-hydroxy D3 and is proven around the world to help with bone strength, immune support, and muscle growth. Research and commercial use for decades show that if you want to get more from your cattle's health and performance, look to the one and only High D. Visit hy-dbeef.com to learn more.
Well, let's dive into today's topic, rumen microbiology, driving production and overall productivity and efficiency. But I guess what makes the rumen microbiologic realm so relevant to ruminant nutrition and rumen production?
Well, it's so relevant because, as most of your listeners would know, that when we feed the animal, we're really feeding the microbial ecosystem. And the ecosystem accounts for the ability of the animal to ingest fibrous feedstuffs and convert them into high quality, humanly desirable and humanly edible protein and energy sources. So the fibrolytic capability is core to the role of the ruminant in human food production. And although we presently pass a lot of starch through these animals, it could be in the future that the role of, well, I will say that in the future, the role of the ruminant is stable because it will always be able to convert not only starches, but also fibrous feeds into humanly edible food.
Now, those, when we dive into rumen microbiology and that fibrolytic, tell us a little bit more just about what that fermentation process looks like, and then what the animal actually uses, what the outputs of that fermentation actually is.
So the calf is born sterile, but shortly, probably as it passes through the birth canal, it's being inoculated, and certainly by 3 days after birth, it already has an active population going on in its rumen. Now, that population is going to change over time, and eventually it acquires sort of a mature state. By the way, the development of this microbial population is somewhat governed by the genomics of the host animal in which it resides. And eventually, then, this population, which is very diverse, consisting of bacteria and methane-producing organisms, which we call now archaea, protozoa, bacteriophages, which are like viruses of bacteria, and fungi, anaerobic fungi. This whole microbial consortium has the ability to degrade cell walls of plants down to the component carbohydrates. The predominant one is glucose. And that glucose then is fermented by a common pathway that's even used in mammalian metabolism down to the level of pyruvate. And from there, the carbon flows to various products depending upon how the hydrogen derived from that catabolism is being used. Typically, that hydrogen is flowing to methane, and when the hydrogen flows to methane, then the products are going to be acetate, propionate, and butyrate, principally, plus a few other short-chain fatty acids, some of which are derived from amino acid catabolism. So those products, acetate, propionate, and butyrate, are called short-chain fatty acids. And they account for about 70% of the metabolizable energy requirement of the host animal. So the fermentation is converting a plant cell wall, which has energy that's inaccessible to the mammalian tissues, into a substrate which is metabolizable by the mammalian tissues. And it's vitally important. In addition, these microorganisms are synthesizing and carrying out their own growth and synthesis of cell biomass, and that cell biomass passes then on down through the omasum to the abomasum, where proteolytic digestion is initiated. Protein digestion is completed in the small intestine. Amino acids arise from that bacterial protein, and those bacterial amino acids or microbial protein amino acids, speaking now about the whole microbiome, They can account for 40 to 100% of the amino acids available to support the host animal.
Which is amazing. We can't do that as people.
We cannot do that as people. There was a paper published, this is going back in time, in 1966 by a man named Virtanen from Finland. He fed a lactating dairy cow a diet that contained only urea and ammonium salts, and he demonstrated that that cow could produce almost 9,000 pounds of milk in a year, kind of a small amount of milk, but she could produce milk based upon consuming a diet that did not contain protein.
So creating protein that's usable for us, for people, for monogastrics, out of things that we can't utilize. It's an amazing system.
It is an amazing animal.
And on top of that, that rumen is also producing B vitamins and other essential nutrients through those fermentations. So we always talk about volatile fatty acids, energy. We talk about protein synthesis and breakdown, how magical that is. But even some of the basic building blocks, apart from amino acids and energy, but vitamin production, things like that we don't have to supplement. That animal produces its own.
That is correct. So as you know, and as many listeners know, we formulate for really protein, nitrogen content in the diet, which expresses crude protein, and it gets a little more sophisticated than that. But protein and energy source, trace minerals, pay attention to the macro minerals, and then vitamins A, D, and E. No B vitamins, no vitamin C.
Well, and if we look back in history of just that, this system is amazing. Just the microbiology of that rumen, what it's able to ferment, what it's able to do, how it's able to be able to feed that host, it is amazing. Now, when we look back in literature, there's a lot of information about modifying that rumen environment. I guess what have been some of the progressions or major breakthroughs in this area of study over the previous decades?
I think the most significant modification of the rumen fermentation involved the recognition and approval and implementation of Monensin feeding in the beef cattle industry. Dr. Art Rahn started on this initiative in 1965 at Lilly Research Labs. In 1968, he and Leo Richardson set up a screen to search for compounds that would modify the rumen fermentation. It was really an insightful kind of screen, and I'm going to say that I believe it was based upon his knowledge of the literature. There was knowledge then already of the importance of propionate as an energy source for the ruminant animal. But he must have also been aware of Mike Woolen's work with the fermentation balance that says that if you start with a substrate, you have to account for all of the carbon and all of the electrons in the products. And so Ron set out, I believe, with this hypothesis that says, I'm going to look for something that will increase the production rate of propionate, because it should result in decreased methane production, and the cumulative effect should be an increase in the metabolizable energy value of the diet. And if that's the case, this should result in an improvement in feed conversion efficiency, which would have economic value. So it was a screen based upon surrounding science that had an application in mind from the beginning. So that's really been the most significant modification that occurs, that has been found and that is still in use with regards to the rumen fermentation modification. A second one, which I don't often see highlighted, but I'm going to highlight it here, is really Earl Bartley's discovery of paloxylene for bloat control. That too is a product that's still around and in use, and it doesn't have the same comprehensive impact as Monensin does, but because bloat is still a mysterious problem, I want to give credit to Earl Bartley in that discovery. After that, I think I would say that the recognition that Megasphaera elsdenii could perhaps play a role in expediting the transition when cattle are being moved onto a high-grain diet, because Megasphaera elsdenii is a lactate consumer, is another nice innovation in the field. It has even less impact than poloxamine, but it still was a pretty clever use of our knowledge of a pure culture's nutrition and metabolism, and then creating a product that could be viable and introduced to an animal on arrival or soon after arrival in a feedlot. Beyond that, the knowledge of the field has just grown immensely, but there haven't been many practical products to arise from all of that knowledge. So sometimes I've been disappointed that we haven't produced more from all the knowledge that we've gained, but we have certainly learned that modifying the rumen is a complex task. And the discovery of Monensin and other similar ionophores was, well, kudos to those discoverers. We haven't matched that in succeeding decades.
No, I agree with you. And before the podcast, we were talking a little bit about Monensin, and I'm really glad you brought that up because it's revolutionized, and it's been something that's been consistent. It's been effective, both on grazing animals as well as feedlot animals to mitigate and moderate some of that fermentation profile. Now, poloxamine, fill in the listeners that may not be as familiar with that particular product and how it interacts with the rumen to reduce some of our frothy bloat concerns, because it's an amazing process as well.
It is. So, as I've already mentioned, methane is one of the gases produced by the rumen fermentation, but the other gas, which is produced in even greater quantities, is carbon dioxide. So those are the two principal gases produced by the normal rumen fermentation. And the animal has a process whereby its ruminal contractions will allow the area in front of the opening, the reticulo-omasal orifice, the cardia, the area at the junction between the esophagus and the reticulum. When the cardia is clear, the gases produced by the fermentation are able to be eructated or belched by the animal. In bloat, a froth or foam forms, and now, even though there are these ruminal contractions which aim to clear the cardia so that eructation can occur, the cardia is not cleared because it senses the presence of this foam touching the tissue, and so the eructation reflex does not occur. But the microorganisms are happily continuing their fermentation, so they just continue to produce gas. The animal cannot release it, and so the balloon of the rumen inflates. Eventually, if that pressure is not released, it shuts off blood flow to the distal, to the rear end of the animal, and the animal is not able to walk. And more importantly, it inflates the body cavity so much that the animal has no space in which to inflate its lungs. So it really dies also of asphyxiation or suffocation, I should say, suffocation. So poloxamine now is this product which is a surfactant, which means that it is a product that when introduced into rumen contents causes a reduction in the surface tension, which is binding the film which creates the envelope in which encapsulates the bubble of gas. And so the surfactant causes the bubble to rupture. And now the rumen contractions can place this gas cap, which resides over the rumen contents, in front of the cardia, and eructation can occur. So the principle is that poloxamine, which Earl Bartley brought to the forefront in the industry causes this froth to be disrupted so that the gas bubbles coalesce into a larger bubble, which is now able to be eructated. And all other strategies to try and treat bloat follow the same principle of trying to reduce surface tension by introducing some kind of a surfactant.
It's super effective, and it's amazing just that. And I've seen it in cannulated animals where they start to get a little froth, and you administer, and it's that froth mat just goes away, releases it into a true, gas bubble, and then it's able to be released and everything else. And it's, I remember in veterinary school, we always talked about finally releasing the gas from the mat and that foam. And it's, if you have a bunch of bubbles, you have to release all of the gas from all those individual bubbles to get back to that fiber mat. And it's, it's an interesting product. And I agree with you. It's been extremely effective at having.
I did a seminar on ruminal bloat when I was back in graduate school, and I contacted Bartley, and he sent me some slides, which I used in my seminar. And one of these slides is a photo of an animal from which the rumen cannula is being removed, and there's this projectile stream of rumen contents. In fact, Majak from British Columbia, who later studied bloat in the 90s, he would measure the severity of bloat by the distance that the rumen contents would be projected when he pulled the plug out of the cattle. He was studying legume bloat.
I have that picture. I have used that picture in a class before. No, I know exactly which picture and slide you're referring to. That's good.
It is a classic.
We've seen some of those revolutions in this area of study. We've had a period, I think, at the time of folks looking into prebiotics, probiotics, different substrates to interact. All with varying activity, I'll say. And now we're at a period of time with our scientific and our technology to be able to use technology to help with scientific advancement has just been astronomically high. Are we at a time that you said you're a little bit disappointed that we haven't come up with anything new? Are we at a time that science is trying to catch up with the technology to be able to make some of these breakthroughs?
Well, I think so. I think the technology is giving us additional insights which were simply not possible to be recognized in prior times. The challenge for the rumen microbiology and ruminant nutrition researchers is to synthesize this understanding into a vision for how to effectively modify the rumen fermentation. And the current societal interest is to reduce methane emissions by the animal. But we all know that while that can be a desirable goal, it won't really matter to the cattle industry until there's a beneficial effect realized by the cattle industry from reducing methane emissions. So an issue right now is that we can do some things to reduce methane emissions by ruminant animals, but it doesn't necessarily manifest itself in improvements in productive efficiency of the animals as we always understood it. So methane is an energy loss of 3 to 12% of the gross energy consumed by the animal. If we reduce methane emissions, then that energy should be available for the host animal to use, and that should manifest itself in improved growth rates in animals or improved feed efficiency. But the methane emission modifiers haven't necessarily consistently produced that sort of benefit. So there's something about this modification that we don't fully understand. And so my answer to your question is that with this increased body of huge information. It now needs to be distilled into a strategy that enables the loop to be closed, whereby there can be a practical economic benefit associated with this new kind of rumen fermentation modification.
Now, is methanogenesis and the loss of overall efficiency within the rumen is one area. I agree that there's opportunity to be able to recoup that energy loss and keep it in the animal to build muscle and fat and bone and all of those things. Are there any other inefficiencies with the rumen fermentation that maybe we haven't been able to capitalize or dive into yet?
Well, there's another one. I mean, now it gets a little further from practicality, I'll say, but there are some inefficiencies. There's a futile cycling of nitrogen that goes on in the rumen, whereby I'm going to say it this way: bacterial biomass is synthesized in the course of doing their work. In the course of the bacteria doing their digestive activity, they grow. Presumably, due to the predatory activity of protozoa, some of those bacteria are ingested and digested by the protozoa, and the protozoa digests that bacterial protein, which is a component of the bacterial cell, and that protein is released as ammonia, and the ammonia then can only be used for the benefit of the animal if it's reincorporated into an amino acid. So there is some futile cycling of nitrogen that goes on in the rumen, which is an inefficiency. On the other hand, so therefore one might say the protozoa are bad. But on the other hand, the protozoa impart a certain stability to the rumen fermentation because they ingest starch granules and thereby mitigate the pH decline or the acidification of the rumen following a starch meal, ingestion of starch by cattle. So the protozoa there are these pros and cons associated with the protozoa. And that's the inefficiency that comes to mind. I can't think of any other inefficiency right now, but you're welcome to remind me if you're thinking of one.
No, I'm not. I was honestly just curious because I'm thinking about new frontiers and areas of potential areas that, for rumen microbiology, it's a very, unique area of study. And it's still there's still so much to discover when you're looking at trillions of cells and how they interact with each other. I figured I'd ask the expert of, hey, if there's any other, low hanging fruit for other people to dive at.
Yeah, I don't think so. I just think that understanding the rumen fermentation helps also in the management of anaerobic digesters. They are a different kind of anaerobic fermentation, but they have many of the same principles. I had a civil engineering student come and do his PhD in my lab, and he worked on the problem of fixing the anaerobic digestion that occurs in sanitary landfills. So sanitary landfills can go sour where they do not produce methane, but they're overloaded by volatile fatty acids. And so he did some work in my lab, and that's one of my most highly cited papers is this work we did to understand the microbiology of organic matter decomposition in sanitary landfills. So the principles that we've learned from studying the rumen have had these other very useful applications. And it will be interesting to see if there are any major new principles that arise given the new technology that is able to be applied to the rumen fermentation.
No, I agree 100%. I think there's still so much opportunity that I hope sometime within my career, I see some of these big advancements. I truly believe we will. I think we're scratching the surface on a deeper level of understanding where we can start interacting with some of these systems more effectively, maybe more targeted. And I think that's fun. I think it's exciting because I know there's opportunity.
Yep, I think so too. And I want to say something for the benefit of the ruminant nutrition community. And it is that through the study of the rumen over the years, and perhaps anaerobic microbiology in general, there is a concept which one of my students taught me, which I think is underappreciated in the field of ruminant nutrition. And this is the concept of an electron tower, or sometimes it's called a redox tower. Have you ever heard of this, AJ?
So I've heard of it. I don't know a ton about it.
All right. I want to say a little bit about this concept because it is a unifying principle that can help one put pieces together in sort of a systematic and logical way. So the redox tower or electron tower is a method for organizing the redox potential of various pairs of various redox pairs. And a redox pair could be a proton and the molecule H2, hydrogen. Or a redox pair could be nitrate and nitrite. Another redox pair, which is very important to all of us mammals, is oxygen and water. So these pairs all have a redox potential assigned to them. That's a little bit of a simplification. But one can organize these values, these redox values, these standard redox values from very negative values to very positive values. And one then can think about those redox pairs that are very high in this tower as having a lot of potential energy, kind of like the water in a water tower or the water behind Hoover Dam. If you think of the water in Hoover Dam and you have a water level that comes up to the dam at a particular point and you poke a hole in the dam that's 10 feet below the surface, well, the water that's going to shoot out of that hole is going to create somewhat of a jet. But if you poke a hole in Hoover Dam that is 1,000 feet below the water surface, the water coming out of that hole is going to be a powerful jet. So the rumen is a very reduced environment, and that's why methane is able to be performed. But when those electrons that would normally flow to methane, if they could flow to oxygen, and oxygen is a very positive number and very low on this organization of the redox tower, there is a lot of potential energy to be realized by those organisms if they can find an oxygen. And if all of that energy that's available to them, they could use that for the synthesis of ATP. And of course, the first limiting nutrient in the rumen is energy. But in the rumen, there's not very much oxygen. There's just a little bit of oxygen that comes in from the arterial blood supply to the rumen wall. And there are some organisms on the rumen wall, and they snap up that oxygen right away. So now you don't have oxygen. Well, what they do have a lot of is CO2. And CO2 can be reduced to methane. But the advantage, the energetic advantage of that is very small, only allows for the synthesis of 1 ATP. On the other hand, if that animal ingests a plant like a corn stalk that has nitrate in it, that nitrate has a much more positive value. It's not as positive as oxygen, but it's pretty far down that tower. Well, those organisms, they just snap up that nitrate. So it gets reduced to nitrite. Well, and that's why we have nitrite toxicity in ruminants. Some animals ingest high sulfate water. That sulfate comes into the rumen and it's a very good acceptor of electrons. And the organisms can make a lot more money because that's a much more, it's not quite as positive as nitrate, but it's a lot more positive than CO2 reduction of methane. So the sulfate gets reduced to sulfide and polioencephalomalacia occurs. So, but in the rumen, those things don't happen very often. The point I want to make is that the redox tower is a way to recognize where these other potential electron acceptors fit and what makes them so advantageous to the rumen microbiome and why they are so rapidly metabolized. And then those products of that reduction, they can cause problems for the animal. But the redox tower is a way to organize all of these kind of seemingly disparate metabolic abnormalities into a theme that is much more easily comprehended.
It's almost easily digested, right?
Yeah.
It's time for our famous three.
Still delegating work to someone who hasn't adapted to AI? You don't need another assistant stuck in 2015. You need a remote AI specialist who can handle inboxes, research, reporting, ops, admin, sales support, and marketing, using tools like ChatGPT, Claude, OpenClaw, and automations. Wise VAs connects you with AI-trained Latin talent in your time zone at up to 80% less than a US AI hire. Schedule your free strategy call at www.wisevas.io.
Well, Dr. Schaefer, this is I've been learning. This has been a fantastic conversation. I hope we get you back onto the show another time to keep diving into some of these concepts. But I know we're getting close to time. But I can't let you go quite yet. There's a part of the Beef Podcast show. We always ask our guests a couple of questions. They're pretty, we talked about it beforehand, but are you ready for the three questions? Okay, so number one, you've been a lifelong learner. What is your go-to, especially now in retirement, but you're still in the industry, you're still consulting. What is your go-to location to find information to stay up to date on science?
Well, in terms of staying up to date on science, I actually go to the Web of Science and I search using keywords to find research articles or review articles that inform me on the question that I have in mind. So I would say the Web of Science.
Yep, Web of Science. Now, we also talked prior to, you do have a resource that you wanted to make sure that you wanted to share with some of the listeners. And I wanted to give you the opportunity to be able to share that.
All right, very good. So with regards to this concept of the electron tower or the redox tower, I have a handout here. This is a handout that's called Simplified Overview of Microbial Metabolism. It's a handout that was developed by Paul Weimer, who is now retired as the research microbiologist from the U.S. Dairy Forage Research Center. He would bring this handout and this information into our ruminant nutritional physiology course, a graduate level course. With Paul's permission, I have the opportunity to distribute this overview of microbial metabolism, which includes a description of the electron tower, as I tried to express it. At this point, the best method for distribution of this handout would be if folks were to contact me.
Absolutely.
By email. And my email is dan@schaefer-beef.com.
Hey, pretty simple. Dan.Schaefer /-beef.
-beef.com.
.com. Fantastic. No, I think that's a great resource, and really wanted to make sure that we mentioned that on the show. Question number two: what are you diving into outside of work? You're at the end of the day, you're still partially retired. What information are you taking in, and how are you taking information outside of animal ag and business, so to speak?
Well, I guess I'm a reader of the Wall Street Journal. Early on in my academic career, I always wished I would have more time to read the Wall Street Journal. So now that I have a bit more time, I like reading the Wall Street Journal, and that's my main information source. That's not animal science or animal science research.
No, fair enough. Wall Street Journal. So question number 3. This one I had fun kind of targeting this question towards you specifically. But what characteristics would you say make a true professional and an excellent colleague?
Well, a true professional to me is someone who is a scholar, meaning that they handle information carefully. They recognize what they know and they recognize what they don't know, and they're willing to admit that. A true professional is also someone who contributes to a pleasant workplace so that the communication among the team members is wholesome and productive and progressive. Those would be 2 key characteristics. Handle information correctly, carefully, and help those around you by looking for opportunities to fill needs.
Which is kind of what you're doing now. You're filling some of those needs with some of your clientele, right?
Yeah. I'm also living out 2 pieces of advice that were given to me by my 2 mentors in graduate school. I just want to mention them. I studied under Marv Bryant at the University of Illinois and Carl Davis, a ruminant nutritionist. Both were excellent. It was the very best combination of mentors for my combined interests in ruminant nutrition and rumen microbiology. Bryant said to me one day, as he was coming out of his office, and at the time he was editor-in-chief of Applied and Environmental Microbiology, and he stopped me in the hallway, and out of the blue, he said to me, "Always be a scholar." Okay, I get that. He's reviewing papers, he's seeing various kinds of scholarship present in those manuscripts, and he's saying, "Always be a scholar." And then Davis one day at noon over lunch said, work with your mind before you work with your hands.
That's fantastic advice. I've never heard it put that way, but I love it.
Yeah, so those are 2 phrases that still live with me, even lived with me as I was preparing for this podcast.
Well, it's excellent words to live by, and it is excellent advice. And I'm thankful you were able to share that, with us and with all the listeners of the Beat Podcast Show. Is there anything else you'd like to leave our listeners with before we sign off?
It's been a pleasure, AJ, to be interviewed by you, and thank you so much.
Dr. Schaefer, thank you so much. It's been a pleasure to have you as a guest on the Beat Podcast Show, brought to you by Wisenetics. If you like what you're listening to, please don't be afraid to share this podcast with friends that you know to be able to share more information far and wide. Again, thank you for listening in. Thank you to our guest, Dr. Dan Schaefer. And until next time, we'll catch you on the next episode. Thank you.
Thank you.